Hello friends, now today I am going to discuss a new topic and the topic is the laser scanning confocal microscopy. As the name suggests, it has a laser beam which scan the sample and finally we get the image uh, with the help of camera. And comes to the history. First of all, it was invented in 1955 by Morvin Muskie, but at that time he used a photo multiplier tube to detect the image, and the helium neon laser is used for the production of laser beam with the help of this inert gases, helium and neon. But further in 1980, there were development of solid state laser and then charged couple device, that is CCD camera, were used. And nowadays there is a multi-photon excitation technique is used for the production of the laser, and high quality high megapixel cameras are used for capturing the image. Now comes to the principle of laser scanning confocal microscopy, that the laser beam penetrate deep into the tissue so that we can analyze the three-dimensional structure. The laser beam having the high frequency value, and the sample is actually covered with the fluorochromes, and as we know that these fluorochromes are getting excited when they are getting some high frequency energy, it will emit out low frequency energy with the high wavelength, with the long wavelength light. And this long wavelength light or the fluorescent light is detected by the detector. Here we can see there is one pinhole is used, so that's why we are called it as a confocal. The pinhole is actually only focus the fluorescent light, and rest of unwanted fluorescent which are scattered here and there is getting eliminated. So due to this we can get a high resolution and with a high magnification image, and this is the basic characteristics of this um, LSCM. Means it has a high resolution, it has a three-dimensional imaging because the laser beam is getting penetrate deep into the sample, so we can get a three-dimensional image also. Multiplexing uh, means different type of color we can also produced with the help of different chromophores or fluorescent materials. Specificity, it is very specific because particular fluorescent dye is added to a particular protein, so we can visualize a particular protein as an image. It is non-destructive in the nature because the fluorescent will not damage any kind of structure of the cell, so we can also analyze the live cell imaging with the help of this LSCM. Now in this slide, there are different parts of LSCM is presented. The basic one is the Z control. We can move the sample uh, by moving the slide. Then there is one objective lens which focus the laser beam to the detector. Then there is one beam splitter which split out the laser beam, and only the fluorescent light travels to the detector. Then there is one pinhole. As we know that pinhole is required to eliminate unwanted fluorescent light, and it will only focus the fluorescent at the center, and that will be detected with the help of camera. So there are different applications of LSCM, like we can apply it for the stem cell research, for fluorescence in situ hybridization, lifetime imaging. So what is the meaning of lifetime imaging? Means the fluorescent material is getting uh, getting dyed, or we can say that there is decay in the fluorescent material over a time. If we apply this fluorescent material to a particular cell, and we can observe the different marker of that particular cell, so we can detect it for a longer time. Then next is the DNA hybridization, then membrane and ion probes. So we can also detect the cell membrane and different ion channels with the help of this fluorescent material. Then bioluminescent proteins. Some proteins are naturally getting giving the color. It is called as bioluminescence. Then we can also take the epitopes for the study of a particular epitope. Now comes to the advantages of LSCM. So as we know that it will gives a high resolution, high magnification, increase depth of field, reduce photo bleaching. So what is the meaning of photo bleaching? Means there is as we know the photo is bleached over a time due to the fluorescent material, but the fluorescent dye which are strongly bound to a particular protein, so we cannot get any type of photo bleaching is there. Then reduced background noise because of that confocal or pinhole technique. We can also analyze live cell imaging, multiplex capability. That means multicolor. The image can be get into the multiple color. It will get it will give us accuracy, and it will give us a 3D picture set. So we can also see the three-dimensional structure of the sample. Now comes to the limitation of LSCM. It is very complex and the instrument is very expensive and the sample preparation is very tedious. Thanks. So this is all about the LSCM. Thanks for watching this video.